Lab 12, the senses. We're going to start with your general senses. And the first figure we have is a figure of your skin model, which you did see and should know from the integument. We have 4 general senses here that we're going to look at, different structures. We have the free nerve ending, which is the yellow, that comes up here and has nothing on the ends of the nerves. Then next to it we have what used to be called the Mesners, but is now called the tactile corpuscle. It has something on the end of the nerves. This is going to be for very light touch. So when there's pressure, it will distort the Mesners or tactile corpuscle, and it will detect the light pressure. Then we have, for deeper pressure, located deeper in the tissue, what used to be called Pacinian corpuscles, but now you can refer to them as lamellated or lamellar. Either name is correct. And this is going to be due to distortion that pushes deeper into the tissue and will distort these. Lamellar means layered. This has numerous layers like an onion. And the fourth of the different general senses we have are wrapped around the hair follicles, and these are called hair follicle receptors, and they will respond to movement of the hair. So if a bug is walking across your arm and hits the hair, it will trigger those responding. Now we have a few slides to look at. 2 of those receptors. We have the tactile corpuscle. Now when you're looking at this, you're going to notice, and it's going to be pointing upward, it's embedded in some dense, irregular connective tissue that is found in the dermis, and this is at the very top of the dermal tissue of the skin. The next one we have is an isolated lamellar corpuscle. Looks like a little red balloon. This would be found deeper into the tissue for deeper pressure. And the last slide we have a picture of is a muscle spindle. Now this is found in your skeletal muscles, and it is a specialized stretch receptor called a proprioceptor. And this is the spindle. It looks like this little wormy structure that's embedded between skeletal muscles. And you should be able to identify the skeletal muscles because we've seen those in the tissue before.